Hello, my name is David Tarkalson. I am a research soil scientist with the Agricultural Research Service in Kimberly, Idaho. And today I want to talk to you about the effects of dairy manure on microbial activity of soils in Idaho. Southern Idaho has seen a drastic increase in dairy cow population over the last 20, 30 years. You can see on the graph that since the mid to late 1980s, up to the present, we've seen this drastic increase in dairy cows. Therefore, we've also seen a drastic increase in the amount of manure that's being applied to land. And we've noticed that there has been some changes in how this soil functions compared to the pre-manure scenario. In 2004, a study was initiated at the ARS research station in Kimberly, Idaho, to assess the impacts of dairy manure on crop production and nutrient cycling. Within the study, there were three different treatments. There was a fertilizer treatment, which was basically a controlled treatment where no manure was applied. This particular treatment had fertilizers applied based upon soil samples and recommendations for the given crop in a given year. The M1 treatment on here is a lower rate of manure. You can see it had a total of 60 dry tons per acre applied over three different applications, 3 different years. The M2 treatment had a total of 106 dry tons per acre added over 6 different application years, from 2004 to 2009. And so we in reality had a gradient of manure application all the way from 0 all the way up to 106 dry tons per acre. These numbers within each of the different colored blocks represent the amount in a given year in dry tons per acre that was applied. So due to scientist changes, retirements, etc., shortly after 2009, not much was done with these plots. They just kind of sat there and were farmed uniformly with with fertilizer recommendations pulled from samples off of the field. But we started noticing some interesting observations within these plots. So this is a sugar beet crop grown in 2004, where you can see outlines of the different treatments, in particular those with and without manure. We have the fertilizer control treatment, and we have an example of the M1 and M2 blocks, where the early season growth of the sugar beets is much more vigorous than that of the no manure fertilizer plots. And so this was noticed in other crops as well. And it started making us think about, well, why is this happening? It's been so many years since manure has been applied. We thought we wouldn't see much of an effect anymore, but that was not the case. And so we pulled soil samples from these plots as well. One thing we analyzed in these soil samples we took was that the spring available nitrogen in the top 2 feet was definitely much greater for the manured plots compared to the fertilizer. And when I say available nitrogen, I'm talking about ammonium nitrogen and nitrate nitrogen. And so even in this case, this would have been 5 years post manure application. At this point in time, there's still a lot more nitrogen being mineralized within these plots. And I guess at the time, this was not expected. We were expecting that after 5 years or so that the soil would come back to some type of equilibrium, as that manure had been mineralized over 5 consecutive years, that we'd have some type of equilibrium that would even out the plots in terms of mineralization and maybe perhaps other factors as well. That was not seen. And so we decided to start some studies on top of these plots to look at some nitrogen management treatments and scenarios. Since we saw so much growth difference, particularly in the sugar beets, we really wanted to look at sugar beets in these plots and see if we can understand whether or not if it was just nitrogen perhaps that was playing into these differences in growth within the plots. And so in 2014, and again 2016, we decided to conduct a study where we basically wanted to supply an adequate amount of nitrogen to the sugar beet crop of around 200 pounds of available nitrogen per acre. And what this ended up doing is it basically ended up giving us similar amounts of available nitrogen. But let me bring up my pointer here real quick. Supply the same amount of available nitrogen for both the fertilizer and the M1 treatment. So you can see that in both years, basically the amount of available nitrogen from the spring soil sample and addition of fertilizer was the same for the M1 and the fertilizer treatment. So they went into the season under normal recommended management of nitrogen on an even playing field. Of course, the M2 treatment, which was the higher manure treatment, had much higher available nitrogen in both 2014 and 2016. 2014 in particular. This shows the data, the root yield data in tons per acre, as well as the sucrose ARS yield in pounds per acre for the different treatments, the fertilizer, the M1 and M2 treatment. We can see that with added manure in both years, that we had an increase in root yield when we went from the fertilizer to the different manure rates. The interesting thing to take away from this is the fact that, remember, we went into this in the spring on an equal footing of available nitrogen between the M1 and the fertilizer treatments. I'm going to bring up my pointer here to make sure you can see that. So the fertilizer root yields versus the M1 root yields are different. The M1 root yields are higher, even though we had the same amount of available nitrogen going into the spring and into the sugar beet cropping year as compared to the fertilizer. So there's something that was going on here that was not explained by initial soil samples for available nitrogen and fertilizer added. So it could be potentially a whole host of things we don't understand. It could be a higher mineralization rate during the early part of the season. However, we know from research that the amount of nitrogen we added in the fertilizer treatment was more than adequate to maximize yields. So this increase, this bump in yield with the same amount of nitrogen available in the spring is not due to nitrogen. It's likely something else. And the same type of increased yield can be seen here. However, we start looking at the sucrose yields. We're now taking into account some quality factors as well that is related to the ability to extract the sugar from the sugar beet crop in the factory. And we can see that in particular in 2014 that we do definitely have an increased sucrose yield with the M1 treatment compared to the control. However, when we get to the high rate, we're starting to see that sucrose concentration going down, particularly in the 2014 year. Did not happen in 2016, but starting to see some quality factors come into effect, particularly with this high manure treatment. Here we see the percent sucrose with the 3 different treatments, and the general trend is that as we add manure, we see a decrease in the percent sucrose. Okay, and in some years that continues to go down as we increase manure. In 2016, it went down slightly with the addition of the M1 treatment, and then the M2 treatment did not see much of a decrease in that particular year compared to 2014. Nevertheless, that adding the manure and both 5 and 7 years post manure application, we're seeing this effect of the manure on sucrose. This graph shows the 2 main quality factors that are analyzed in the amalgamated growing area. That is root nitrate concentration as well as the conductivity of the root itself. And we can see the data shows us as we increase our overall cumulative manure application rates, we're seeing an increase in both the nitrate and the conductivity, indicating that quality is decreasing as we increase manure application rates. However, we do see at the highest manure application rate, we see the highest decrease in quality. So I want to bring you back to the beginning of the presentation and the study setup to drive home a point here. The amount of available nitrogen in the spring in both 2016 and 2014 was basically the same between both the fertilizer and the M1 treatment. It was only the M2 treatment that had excess of actual nitrogen from the soil test. But even with the same amount of available nitrogen as determined by a soil test and fertilizer application, we still see that with our sucrose yield, that it increased between the fertilizer and the M1 treatment. So there's something going on here that's not being accounted for with just available nitrogen and fertilizer added in the spring. There's other factors involved besides just that particular measurement and accountability of nitrogen that we typically use for recommendations. You can also see that the sucrose is decreasing between those two treatments as well. This is a very basic description of the nitrogen cycle. And I want to show you this to really show you what happens when we don't understand a big part of the system and how it impacts crop production. We can see that the constituents in yellow, the nitrate and the ammonium, are the two different forms of nitrogen that are available to our growing crop. And that's what we sample in the spring, how much is actually sitting in the soil, and we base our recommendations around that concentration in a sense, and how much fertilizer we got to add. What supplies that is this conversion of soil organic nitrogen to ammonium and then onto nitrate. The problem is this whole area of mineralization, microbial activity, what the actual microbes are doing within the system, both related to nutrients and not related to nutrients, we don't really have a good handle on it. It really is kind of a cloudy situation. And without understanding this, it really hinders our ability to better manage, in this case, the nitrogen cycle. But there's also other components that are part of this we don't really understand. It's not all just nutrients. They are probably a part or a big chunk of the scenario of the puzzle, but we don't understand the other things that are going on. We've basically changed the microbiome potentially within this soil. And what other effects does that have that may not be related to nutrients? Is there effects on soil physical properties we don't necessarily understand very well? And what about all the other things we haven't even asked the question about yet? In the spring of 2020, we went to these plots, all the plots, the F, M1, M2 plots, and we sampled them from 0 to 6 and 6 to 12 inches to assess the soil microbial activity on these soils. It's important to remember this is 11 years post manure application. So manure had not been applied in the past 11 years. We're looking at the residual effects of these manure applications on the soil microbial activity. To look at this activity, we analyzed for 4 different soil enzymes: glucosidase, glucosaminidase, phosphomonoesterase, and aryl sulfatase. We also analyzed for ACE protein and nitrous oxide emissions. This slide really gives more detail regarding the analysis and exactly what each of these enzymes and what each of these different analyses are assessing. So to kind of summarize in general what each of these are doing, I've put those in red and underlined them. So we're looking at both at cellular energy related processes, mineralization of nitrogen, phosphorus, and sulfur. The ACE protein really is a general in mineralization type analysis, looking at the amounts of protein-like substances that are present within the soil organic matter fraction, and also nitrous oxide emissions, which is nitrous oxide is a end product or intermediate product in the nitrification and denitrification processes within the nitrogen cycle, and are microbially driven. So all of these really are a general measurement of of how much microbial activity is occurring in the soil at a given time. These graphs show the 4 different soil enzymes that we measured at both the, let me bring up my pointer here, at both the 0 to 6 and 6 to 12 inches. Just the concentration of these within the different treatments, the F fertilizer control, M1 and M2 treatments. And you can see that across all of these enzymes at both depths, we are still seeing increased activity or increased concentrations of these enzymes 11 years post manure application. So that manure application has had a significant effect on the microbial community, its activity 11 years post manure application. We also here have the ACE protein concentrations for the 2 different depths, as well as nitrous oxide emissions at 2 different depths as well. And the same type of summary can be drawn here as well, that with manure applications, we're seeing increased ACE protein concentrations and also increased nitrous oxide emissions compared to the control 11 years post manure application. So what do we know from all of this analysis I just presented? What can we conclude from it? Well, what we do know is that manure, unlike the other manure additions, if you want to extrapolate that far, influence the soil microbiome. And in this case, increase the activity of the soil microbiome after manure applications, at least up to a decade post manure application. But what we still don't know is the important stuff. And we're starting to maybe understand this a little bit, but what we want to do is be able to take this data and all this information we're gathering from research and to model the actual nutrient mineralization, model some of the non-nutrient effects, model all the interactions between the factors so we can actually base management upon that data we get. We're not quite there yet. We also have lots of things we want to know that we just don't know the answer to, and sometimes we don't even know what question to answer. So we still have a long ways to go. This is really the stuff that's hidden from us and really obscure, as I mentioned before in the nitrogen cycle slide. You can look at it similar to this picture. This is an iceberg. You can see the diver here. And we all know that icebergs are very large, and most of the mass, in some cases 8 to 92%, 8% of that mass is above the water, around 92% of that is below the water. And so if we want to make this an analogy to what we're talking about today, what we do know is a very small fraction of what we don't know. But what we don't know is what we need to know. It really is what we need to know so we can make better management decisions. And so we got to get there. So to do this, at the USDA Agriculture Research Service in Kimberly, Idaho, and the scientists that work here have many studies. I just listed 4 here in which we're trying to better understand and get a better understanding of what we don't know, or exposing more of this iceberg, that is the soil microbiome. And so it's our hope that throughout the next many years that we'll have a better understanding of how to understand this system, therefore better able to make management decisions based upon a greater knowledge of the entire system. Thank you for letting me be able to present this today. Please contact me at the email address below if you have any questions. I'd love to talk to you. Thank you.